everybody. Good afternoon. I think we'll get started. So I'll be giving a talk on some of the research that we do in our lab, which is essentially soft matter manufacturing and also living tissue manufacturing. And this includes my work as well as work of some previous students in the lab. And towards the end of the class, we'll talk about the exam and grading, and I have the graded exams here that you can all pick up. So I work in the lab of Dr. Thomas Angelini. It's essentially a soft matter collective cell mechanics lab, and the lab website is down there if you want to check out some of our publications and research work. So a little bit of background. So when we want to think about manufacturing soft materials, especially like bioprinting, where we print like cells into little tissue models, we need to think about the stresses that are in place and the strains, right? And if you look at the figure on the top right, if we want to say a material is smushed under its own weight, let's call that like 10% strain. For really soft materials, for example, tissues, which are like in the range of 1 kilopascal, to do a 10% strain, you would just need a height of like 10 millimeters. So if you try to print like a soft 10 millimeter pillar, it would just smush under its own weight. And that gets even more dramatic if we then move to slender beams. It goes down by a factor of 10. For a 10% strain, you'd literally just need a millimeter height of beam, and then it would just sort of buckle under its own weight. So precise design and manufacturing of a lot of these freestanding soft structures is a big challenge because of this. And this is the reason why we need bones in our body to hold up the soft tissue together, right? So when we want to like print soft materials, if we don't have like scaffolds or like bones, then what is the option? So that's what our lab looks into. So here's a lot of examples of 3D printing soft matter in our lab. So on the top left, you would see cell suspensions being printed into like little tissues. Here in this case, it's like a disc-shaped tissue. Here it's picking and placing like individual cells. The ones at the bottom are essentially printing PDMS silicone into another type of emulsion support material. And we can print like really precise structures. Like for example, this is a brain aneurysm model, and we can get down to like precisions of 8 microns in that case. And we can also print like just aqueous polymer suspensions. So this jellyfish here is just being printed out of an aqueous polymer suspension and into this material that we call microgels, which I'll talk about later. So this is just sort of an example of how precise manufacturing of soft materials can work. Yeah. Yeah, these are individual cells. Oh, so we use in the scale of cells, we do it on top of a microscope. So we have a confocal microscope where we can actually image while we print, and that's where we do imaging. And if you see the scale, yeah, like a single cell scale is like 10 microns, which is like even way lesser than even a diameter of a human hair. So some of the things that I wanted to talk about, I don't know if I can go over everything given the time, but I'll probably touch upon the first 2 or 3 topics and go from there. So we call this method the embedded 3D printing. So we need to make a support material which is also soft, and it also holds soft inks in place when we print into them. So there are considerations of yield stresses and how the ink would behave when we print into these sort of support materials. So say, for example, we just take a liquid and then we try to print another liquid medium in there. It's just going to break up into droplets, right? It's not going to hold its shape together. Say you have a really stiff support material, maybe like hard tofu or a really stiff jello or something. You try to print into that, you're going to form crevices because the yield stress is too high in that case. So then what we need is somewhere in between, which is a material that behaves like a solid at low shear, but at high shear rates, it behaves like a fluid. So then you can actually print into them, and once you print into them, it basically holds the shape together. So that's essentially the idea of embedded 3D printing in our lab. And the support material that we do can be in the form of microgels or emulsions. Microgels are essentially like microscopic polymer particles that are like packed together, and they can be swollen in different types of liquid media. Like it can be ethanol. Hand sanitizer is a great example. Hand sanitizer is actually a pack of microgels swollen in ethanol. And the idea is, in the macroscopic scale, the idea is kind of like a ball pit. So if you have a ball pit, a kid is in there. If you just don't do anything, the kid will be supported by the balls. But if you just start moving around, he can actually swim in the ball pit, right? So that's the general idea of how this embedded printing works. So when we do this sort of fluid and fluid printing, or fluid and like soft solid-like material printing, there is a consideration of interfacial instability that comes in. So in our lab in the past, students tried like printing different types of inks into different types of support material. For example, here it was like oil printed into an aqueous gel. Here it's an organic micelle printed into an aqueous gel, organic microgels printed into aqueous gels, and then solid beams in Maxwell fluids. So if anyone is interested, you can look up that paper. There's a lot of information over there on how this printing works and the mechanics works for this. But as a general idea, when we make these sort of soft materials, we want to characterize the material properties. And in our lab, we use this technique called shear rheology. A rheometer is essentially a tool where you have like this shear oscillation capability. So you can just sort of sandwich your material between two plates, shear it, and then by shearing it, you can either do like oscillatory shear or unidirectional shear. And by the mode of shear, you can figure out what are the material properties. So the two most important properties we look at are modulus and yield stress. So in the regime of soft fluids, like in shear rheology especially, modulus is considered as a complex modulus. So then the real part is considered as a solid-like behavior, which is G prime, and it's called storage modulus. And then the imaginary part is considered as a viscous component, which is a viscous modulus, that is G double prime. So for example, if we look at the plots on the rightmost corner, if there is like a weak frequency dependence for storage modulus, it's considered as an elastic solid. And also if the storage modulus is above loss modulus, it's considered an elastic solid. And by doing unidirectional shear, you can back out shear stress over shear rate graphs. And from this, when this shear stress over shear rate graphs plateau, that plateau stress is what you would call the yield stress of these kind of materials. So this is sort of a general way of characterizing soft materials. And it's a little bit different in how we think about like modulus. Like in metals, it's just Young's modulus, right? But in soft materials, you have this solid component and viscous component that comes into play. Like I was saying earlier, this is just examples of different types of inks printed into different types of support material. So here, for example, it's either mineral oil or organic micelle printed into an aqueous solution. And there are considerations where you need to look at what regime of yield stress and what regime of diameters you can print to get a stable structure. Like for example, if it's a really large diameter and really low yield stress, you're going to break up the printed material. So this was sort of mapped out in the work that I talked about earlier. And you can figure out what diameters you can print without breaking up your printed structure. In the same way, this is sort of a solid beam in a solid support. Again, this is like solid-like beam in a solid-like support. Essentially, it's an organic microgel printed into an aqueous microgel. And we can again map out this whole regime of where we can get stable structures and how we can carefully choose the yield stresses of the support material. And then there's this fluids called Maxwell fluids. And again, if we try to print these sort of solid-like soft gels into these Maxwell fluids, there are other problems that start creeping up, like coiling of the ends and undulations of the printed structures. And the stable window is really short. It's just like a really narrow window where you can get like stable, good structures. So general lesson here was just to not go for Maxwell fluids when we try to do this sort of 3D printing. And another sort of limitation when we do this sort of soft material 3D printing is this plastocapillary length, which is essentially a ratio of surface tension to your yield stress. And as a general rule, the idea is to not print any features that are smaller than the plastocapillary length for any material. So in this graph, you can see like a whole range of materials from, again, like metals, polymers, all the way down to like soft materials like microgels and hydrogels. So, so in order to print fine structures, we can either increase yield stress or decrease interfacial tension. So, and we choose the second approach where we try to decrease interfacial tension and then print like stable structures. So in terms of characterizing printing, this is sort of a general way where you would print like simple structures like beams, and then you can, from the fluid continuity equation, you can back out what flow rate you need, what velocities you need to move the needle to in order to get feature size that you're targeting. And these are sort of very well characterized at this point in several studies and even in our lab. So we, in this particular case, there was a whole range of flow rates printed, like 3 microliters per hour is like extremely slow, versus 5,000 microliters per hour is fairly fast. And then the entire range of printed structures fall within this this nice relation, which is just governed by fluid continuity. So that's just sort of a general background of what are the considerations we need to do when we think about printing soft materials, which is just very different from additive manufacturing of, say, polymers, right? So we'll move on to some bioprinting work. So this was one of the works done by a past student in our lab where I also contributed a little bit. So this is essentially cell-generated forces that buckle and destroy printed structures. So the way this works is we try to print this cell type called 3D3 fibroblasts. Fibroblasts are this cell type that is just found in abundance in the body. It's just there in connective tissue, in the skins, tendons. And these fibroblast cells are known to grab onto like matrix. And the human body also has like this matrices, which is collagen is one of them, laminin, fibrin. There are different types of these fibrous structures in the body. And this fibroblast is known to just sort of grab onto these extracellular matrices. And here you can see like a sort of a time lapse of how they sort of just reach out and grab and contract. So a couple of previous students like Cameron Morley and Tori Ellison, they printed these little beams, which are just mixtures of these 3D3 cells with collagen. And then they observed buckling of the beams. So, which was just really neat. And the buckling seemed to be controlled by your aspect ratio, the diameter to your length ratio. And these were again printed into these microgels, which were just sort of swollen in like a liquid growth media that the cells need to proliferate, right? So because there were beam buckling, they went down to characterize just sort of beam mechanics. In this case, the difference is it's like a beam embedded in another material with an elastic modulus. You can kind of think of it as a mechanics of steel beams embedded in concrete, except in this case the beams and the concrete are like both soft. It's not really like as stiff as metals, right? So, but you can sort of write down the fundamental equations and back out critical stress that you need to buckle these beams, and also like critical diameters that where the buckles would beam or like the beams would buckle or like stay stable. So by doing this entire range of diameters and different types of beam printing, the wavelength of this buckling was characterized using Euler-Bernoulli theory, which is very similar to, again, mechanics of beam supported on an elastic substrate. So again, this follows this nice little trend. Again, this is not, the data was not fitted, this is just sort of raw data, and they all sort of lie on this nice curve. So it was just found that the Euler-Bernoulli theory predicts this data of undulation of these beams with essentially no fitting parameters. So, and from here, they essentially mapped out onto mapped out this buckling behavior in a phase diagram. Like what if we say increase the number of cells versus we increase the aspect ratio of the beams, and how does that affect the behavior, right? So we can either characterize it as buckled beams or like stable beams. In some cases it was not possible to determine it, so it was just undetermined. And then we can figure out like what threshold, volume fraction, and aspect ratio you need so that you will get stable structures instead of like buckling structures. So essentially the idea is fatter beams, they won't be able to buckle. Also, if you have really low number of cells, they won't be able to buckle. There is a threshold number of cells that are needed to be able to contract and buckle these beams. And then from here, they also looked at varying the yield stress of the support material. We talked about yield stress as one of the criterions, right? Like the support material yield stress needs to be sort of low enough that you can print into it, but not so high that it will, that you won't be able to, it'll form crevices and stuff like that. So you can also map out the shear modulus of your surrounding material versus the concentration of the matrix in your beams. And again, threshold was detected for like what shear modulus we need and what collagen concentrations are needed in order to get stable structures and in order to not have these printed structures buckle. So then from here, there was a certain condition where they saw that the beam started breaking up, and that was pretty interesting, where it was essentially in this low to mid range of collagen and then about mid range of shear modulus. And then because the beam started failing, they started studying failure mechanics and understanding how using these failure mechanics to understand how cells generate stresses. And then this sort of relation was determined where the stress generated by the cell is modulus to the power half. And then they did a whole bunch of experiments with buckling of beams, contraction of beams, and sort of backed out this relation. So it was just sort of a fundamental study of what we need to consider when we just want to print. Really, like the beam is the most simple structure that you would think of when you start thinking about like making something, right? So from here we moved on to a little more complicated tissue models, and this is where a lot of my work has been for the past few years. So we call these the minimal bioprinted tissue models. And why this is important is because cells behave very different in 3 dimensions versus 2 dimensions. It's just studied over and over for like several decades now. What you see here is actually a gene expression panel that was done back in 2004. And you can see the colors are like just anti-correlated between 3D and 2D. It just shows it just says that the cells just behave really different in 3D. And we need to understand how cells behave in 3D if we want to understand how tissues behave, and then eventually try to build organs and stuff like that. So so in our lab, like I said, we use these little aqueous microgels to print cell suspensions. So here you can see sort of an annulus shape printed out of these cells that are called MCF10A cells. And then here there are MDCK cells, again printed like in a disc shape. We can also print really precise structures, as you can see, just compared to a push pin. But we chose to just focus on minimal structures without getting too complicated, just to sort of understand how cells behave in 3 dimensions, right? And the length scales we're talking about here are extremely small. This is just a grain of rice, and we're talking about structures like that. So essentially just like 2 millimeters wide, a couple of microns thick. These particular structures were not printed out of cells. These were like just sort of fluorescent particles, but the idea was to just demonstrate the printing precision. And this is also like modeled out of neural crest development. So I was talking about microgels, right? So for all of these work, what enables us to do this is these sort of packed soft particles, swollen in this case it's not PBS, it'll be the cell growth media itself that the cells need to proliferate. They're made of polyethylene glycol in an inverse emulsion polymerization method. And here is just a microscopy image of how these microgels look. It's just sort of packed, soft, spherical particles. And they have sort of an average diameter of 8 to 10 microns and a distribution of about 5 to 40 microns. We were talking about rheology, so we again characterize these materials because we synthesize them in the lab. We need to understand what's the modulus, what's the yield stress. So we did a whole bunch of shear rheology on these polyethylene glycol microgels, and then we found that at about 4.8 to 5%, where you have a yield stress of say a couple of pascals, is a sweet spot to be able to print these cell suspensions and like little tissue models. Again, these are like elastic solids at low shear rates, but they behave like fluid like at high shear rates. So just to show that when we make something like this, we need to sort of characterize and understand how we can precisely print what we want to print. So one of the models that we tested this on is a human liver tissue model. So we 3D printed a whole bunch of these disc-shaped tissues, so to speak. We started off with like single cells, like just liver cells, and then added on more types of cells. Essentially, there are about 30,000 cells in one of these little tissues. Each tissue is just like about 2 millimeters in diameter, so pretty tiny. And again, cells need this liquid media to stay alive, so we exchange the liquid media every 2 to 3 days. And then when we exchange them, we can actually collect the media and the tissues and analyze them for function. Before I get to the function, I'll talk about just manufacturing. So when we print these, we again characterize like shapes of printed structures, and we call this like metrology characterization essentially. So in this particular case, we just printed like discs of a whole range of diameters, and we can sort of predict what diameter it's going to be based on the flow rate and velocity, the fluid continuity that we mentioned earlier, and we can sort of figure out how precise our manufacturing is. And in this particular case of liver models, we found that our manufacturing was pretty good within an error of like 4.2%, and maximum error in some cases was about 13%. Yeah, so but we got pretty close to what would be an ideal manufacturing condition. And from here, we can analyze function, right? Like when I said, we can actually take off the fluid and tissues and analyze them over time. And here is actually a huge gene profile panel done from these extracted printed tissues. And we can see that, so Hepatopac is actually one of the commercially available, they call it a patterned microstructure model for livers. And ours was much better, and even compared to a human liver, we were really close in terms of how these tissues behaved as a liver. So just goes to show that simple models, just sort of carefully thought about and printed, can actually get you functional tissues. So another example is immunotherapy, where it's applied to cancer. So in this case, a previous student, Cam, he printed these little boluses of tumors and then surrounded by rings of T cells. And we can see over time the T cells just sort of migrate towards the tumor and start attacking them, and the tumor just sort of reduces in volume over time. And of course, we can go and analyze sort of how the T cells migrate and how it's related to diffusion kinetics of these chemicals called cytokines, which the T cells use to sense the tumor. And then sort of following from there, I printed these. There's this really notorious cancer called ependymoma, which is sort of really hard to target. So then we collaborated with the UF Medical School, printed these little tumoroids of ependymoma surrounded by trained or untrained T cells, and we can see like over time, just a lot more T cells migrate towards the tumor, and the tumor itself starts shrinking over time. And in some cases, the T cells even like pick on tumor cells and literally like rip it apart. So it was pretty cool to be able to see that. Let me see. All right. So from here we can analyze this data, right? So we can actually, we take these 3D stacks, put them in MATLAB, segment the different types of cells, and we can say we can quantify how many T cells actually get closer to the tumor, and over time how the number of T cells increase. In this case, we found that the trained T cells just sort of increased in number over time compared to the untrained T cells, which almost showed no activity. And then we also saw the proportion of those immune cells which are associated with the tumor was also much higher in trained cells as compared to like untrained immune cells. So this is sort of work in progress. It was a preliminary experiment. The one thing we also found was all the tumors shrunk over time, which we are trying to figure out why that happens. Essentially, in the untrained case, the tumor should not have shrunk, but it could be like culture conditions or a lot of different factors, right? So we're still working on this. Another cancer model that I worked on is this glioblastoma, again printed as little boluses. I'm hoping the movie would play. If not, I don't know. Anyway, again, these were like boluses of these glioblastoma printed with like conditioned and unconditioned T cells, unconditioned actually immune cells derived from bone marrow. And then we took these 3D stacks of images in confocal microscopes, processed it, and figured out that in the conditioned case, over time, I wish this movie would play, but essentially what happened was a lot of T cells started migrating towards the tumor, and the tumor itself was just like going bonkers. It was literally like moving around trying to like get out of the annulus, and it was pretty crazy to see that. And in the case of unconditioned immune cells, it was none of that. It was just like sitting around pretty much doing nothing, maybe a little bit of motion. So yeah, that was just sort of examples of how we could just print like very simple structures, but actually study in this case even like diseased models, right? These are like the medical school is working on like training immune cells to attack cancer, and we can actually test them in the lab. And eventually, if this method is proven, then we can use like patient cells to be able to test over a course of say chemotherapy, right, to see how they're responding to treatment. So this is a very powerful method that we are sort of demonstrating here. And there was another work that I did, which is how we can stabilize soft structures without messing around too much with the soft material. And in this case, I made like a composite material of the collagen matrix with these spherical microgels, and they showed like really cool behaviors where when we mixed the two of these materials together, the combined modulus of this composite jumped like 10 times as compared to what the individual components would have. And that was kind of unseen in any soft materials so far. So essentially, in this, the idea is to combine the nonlinear behaviors of biopolymers versus the reconfigurable nature of microgels. So biopolymers like collagen or any other biopolymer, right? Usually they are known to like stiffen. If you just train them, they just stiffen, and that is considered like a nonlinear behavior in terms of like the mechanics of it. The reconfigurability of microgels, meaning it's just like a pack of spheres. So when you shear them, they don't really get destroyed or stiffened, but then the pack of spheres just rearrange, right? And we call that reconfigurability. And here we are sort of putting those two together to make these composite materials. And like I said, we were able to do a bunch of rheology tests on these composite materials and figure out a relation for the storage modulus over the concentration of microgels and collagen. And they sort of strongly depend on collagen concentration and a little bit weaker on the microgel concentrations. So then we also wanted to test how this nonlinear behavior would work. So we strained the sample, hold the sample at a particular strain, and just sort of did oscillations at that pre-strained state to measure what the modulus would be, and then again ram the strain back down, and again measure the modulus. So when we did this, there was this really cool behavior where the stress reversed. It's like the stress went in the other direction, which was just really interesting to us. And it happens because we can think of this composite material as like a bunch of fibrous networks with the porous space filled with these soft spheres. So when you sort of shear them, the collagen would strain stiffen, and then when you go back to your zero strain, the microgels, because of the reconfigurability, they'll resist your going back to zero strain. So then what happens is it applies an opposite force on your tool, and you start measuring like negative stresses. So which demonstrated this idea of shape reconfigurability, or like shape memory. I don't want to call it shape memory because this is not like a traditional shape memory alloy, but still it's like tin foil. If you just sort of bend it, it will just hold that shape for a really, really long time. So so we call that shape programmability, where I just tested, I casted sheets out of these composite material, just bent it around, and it just held its shape for like sometimes even several days. So yeah, like 13 days in the case of the bottom little bridge kind of structure. So this is an example of how we can tailor specific properties of, say, biopolymers or like soft materials to make even cooler, like stiffer soft materials. In this case, this material, because of the shape programmability and stiffness, we should be able to like 3D print. Here I printed like a hairpin shape, and it held its shape for again a long time. So yeah, just another example of how we can do composite materials out of these soft structures. So one last project. This was not my project, but another student in our lab did this. But he was working on silicon 3D printing. Interfacial tension plays a role here. I wouldn't go too much into that right now. And we talked about the considerations of instabilities earlier. So he essentially printed different types of silicon materials into these emulsions, and we were able to show that and they have this ultra low interfacial tension between the ink and the support material, and that's really crucial to be able to print say silicon materials into this sort of silicon oil emulsions. And again, the paper references over there if you want to read up more, but this is an example of a whole array of structures that he was able to print using these silicon materials. I was showing earlier the brain aneurysm model. So like I said, we got down to really precise aneurysm models, almost 8 microns precision. So he got this sort of patient data from medical school, 3D printed based on the model. Again, sent it back to the medical school. They did a CT scan of the printed structure and overlaid it on top of the original patient model, and sort of did an error analysis and showed that the manufacturing was really precise. Again, down to 4% or less error between the actual and the printed one. He also printed like heart valve models. This is supposed to be a movie again, but which is not showing. But yeah, he was able to print these little heart valve models and pump fluid through, and it actually behaved like a real heart valve. So I'm hoping this would, yeah, there you go. So this is just how like a time lapse of printing one of these brain aneurysm structures. Again, sort of really complicated structure, which normally you wouldn't be able to print with a soft material just without any support. If it's like free hanging, it would just like collapse. But with this sort of emulsion support material, we're able to print this aneurysm model. And again, this is a render of the printed structure itself, and later on, yeah, compared with the CT scan, and then sort of overlaid together to see how precise our manufacturing can get. So yeah, in the CT scan, you can also like slice through. So yeah, if you take a CT scan, just because you have a really cool 3D structure, it doesn't mean it's precise. So you can actually like slice through different planes, and then you can see the little walls of the tubular structures. And that's how we show that the manufacturing is precise. So, and this is, oh yeah, here's the video of the heart valve, if you want to, hopefully it plays. I guess not. Anyway, so yeah, that's just sort of different examples of how what considerations are needed when we think about soft material 3D printing, particularly printing cells and into like tissue models. And with sort of careful selection of ink and support material, we can print these really functional tissues even. And there's a whole bunch of physical problems and research yet to be done. So this is also a cool field if you guys are looking for doing research in this. So yeah, and if you guys have any questions, we can talk about it after class. But yeah, if you guys have any questions right now, I would take it, and then we can move on to like exams and stuff. Yeah. Yeah. Was that? Not part of Dr. Sawyer's lab, but I'm part of Dr. Angelini's lab. We used to work in collaboration with their lab, but now their lab moved to, I think, UCF. So, yeah, I'm part of Dr. Thomas Angelini's lab. All right, so if there are no questions, let me pull up information about exam 1, and then you all can pick up the graded exams as well after class. So, hopefully that shows up. There we go. All right, yeah, so exam 1 has been graded now. I'll be releasing grades after class. So the grading policy we did was, it was 4 points for most of the problems, 2 points for if it was just like yes or no questions. Again, partial points were given just based off of if an understanding of how to solve the problem was like demonstrated, if some working was demonstrated. There's a 1-week regrading policy, so you all can visit me during office hours. It's Tuesdays and Thursdays, 3 to 4 p.m. I've booked a room in Wright Union just because the Wertheim building is usually with card access and you may not be able to enter. So we'll meet at Wright Union, the 3rd floor. There's an office space, office number 3106 during office hours. And yeah, so on-campus students, whoever is in class right now, we can pick up the exams now. If you have not picked up your exams in class today for the camera, please do so during office hours, again, Tuesdays and Thursdays. Edge students, please follow the 1-week policy, and you can contact me first if you have any questions, and then if you have further questions, you can contact Dr. Greenslit for regrading. So that's all I had.